Good evening. Hi, good evening, Byron bhai. Yeah, we'll start the lecture. You can see my screen, right? Yes.
Yes, sir.
Yeah. Sham Kumar has also joined. Good evening, Sham. Okay, so welcome to the course, week 4. So this is bioengineering course wherein Professor Sanjeev Srivastava is talking about how we can integrate biology with medicine as well as engineering with certain new technologies which are coming into picture. That is because of engineering in the field of biology and medicine. So in terms of week 4, what Sir has majorly gone through uh, was about Mendel's law itself, and so in the week 4 lecture, that is from lecture 16 to lecture 20. In lecture 16, Sir majorly talked about different laws of what Mendel came up with, and how did he exactly study those laws using what kind of plant and how he came up with inheritance ratios, like how inheritance takes place from our parent generation to next generation. So he tried to study those. So basically, he was a keen observer and he tried to study all those stuff. He was not in an intention of studying inheritance, but he just wanted to study how things are happening in nature. Then he later came up with, like, ended up in inheritance. So with inheritance, he found how inheritance happens, and later he was called as father of genetics. And in lecture 17, Sir majorly dealt with different problems on genetics. Then on lecture 18, he started with there are so Mendel, whatever Mendel saw in his experiments, so he has certain way of inheritance, but there are various characters or characteristics in us and in other organisms wherein there is a deviation from Mendelian genetics. So the plant he studied, it followed the laws which Mendel created, but there were some other organisms which didn't follow the Mendelian genetics. So there are certain deviations from Mendelian genetics, or there are some certain traits which he didn't observe, and those traits had deviation from those laws. And Sir talked about that in the 18th lecture. In 19th lecture, how genetic rules can be applied with probability. So instead of using the Punnett square method and all of those stuff, you can use probability to just find out answers and how exactly inheritance might take place. You can use probability for that. And in lecture 20, there was a talk by Dr. Jayanti Sastry. So I hope you all have gone through all of these lectures. Have you all gone through these lectures? Yes. Yes, Sham and Barunda. Have you all gone through these lectures early, or like you haven't gone through it yet? Yes, no? Yes, I couldn't hear you. Hello. You can write in the chat box if not by unmuting yourself. Okay, you have already gone through the lectures. So what I'll do is I'll do a quick revision of all 4 lectures, and we'll try and solve certain problems. And there were already solved examples in the lectures as well. We'll also go through them. So if you are having any kind of doubts, then we can accordingly discuss regarding that. So I'll go through it as fast as possible. And if you're not getting it, you can interrupt me in between so that I'll explain you a bit in detail about that. So what is a genotype? It's an organism full hereditary information, whereas phenotype is the one which we actually observe, the external feature, that's the phenotype. So in terms of genotypic phenotypic ratio, so what Mendel studied was he took a pea plant, and using that pea plant he saw different variations. So you can see uh, in terms of phenotypic ratio means external features. So externally you can see there are 3 purple and 1 white. So phenotypic ratio should be 3:1. But if you see the phenotypic one, this the capital P is 1, whereas capital P small p are 2. whereas small p are 1. So this genotypic ratio should be 1 is to 2 is to 1, right? Ideally. So genotypic is 1 is to 2 is to 1, and whereas phenotypic is 3 is to 1. So that's how he was trying to predict. So how, although externally it looks the same, so how did he determine whether this purple flowers are having capital P as a genotype or a capital P small p as a genotype? Any idea? How did he know that this specific flowers had the genotype of capital P or capital P small p? Yes, as you all have already gone through the lectures, any idea? How did he know that? No? Okay, it's because so he understood that by doing test crosses. So we'll come up with that. We'll come to that test cross. So what? So in order to understand certain terms, so gene is something which is a sequence of nucleotides that you already know, and alleles are different forms of gene itself. So one specific gene can have two types of alleles. So there are two copies. So each organism harbor two copies of a gene. So that two copies are nothing but we call it as alleles. And alternative variations of gene account for variation, which are called as alleles. So that alternative variation of genes are, so it's the same gene, but it kinds of expresses differently. So that is allele for a purple flower. If this is allele for a white flower, it's at the same locus, same. So each organism harbor two copies of a gene. So this the same gene, but this is allele for a purple color and this is allele for a white color. So dominant allele has an effect over the recessive one. So that's what you are saying. There is alternative variations of genes which account for variations, and they are called as allele. So these are the variation. Purple and white is the variation. It is the same pair of chromosome two, but there is one, as there are two copies of it, one of it is act as a dominant one and one of acts as a recessive one, which codes for certain different either purple color or white color in this example. So if you can see, so DNA with this nucleotide sequence has only one nucleotide change. If you see it, so whole gene is the same. Or if you look at the other nucleotides, it's the same, but there is only one difference. Through a series of steps, this DNA sequence result in a production of an enzyme that helps in synthesizing purple pigment. So after like this gene will become RNA, then RNA will become protein. So that because of this sequence, that protein helps in so basically it produces certain enzyme which helps in production of purple pigment and the flower turns out to be purple, and if that is not there, this DNA sequence result in the absence of that enzyme. So if this is the sequence, so there is absence of that enzyme, and that's the reason it results into white color. So, in order to give a brief introduction of Mendel, so he was a monk. At the age of 21, he entered this monastery and he tried to study whether in that specific monastery pursued 2 years of study in physics and chemistry. So he was not a biologist, neither a genetics person, but he was loving. He generally likes the nature, and he was observing that specific nature and tried to study the breeding in garden pea plant and during his experiment in the year 1857, and in 3 years he tried to provide the laws of inheritance to define the genetic principles. So in those 3 years he tried to study different traits. All of those traits we'll discuss. So he tried to answer these questions like what are heritable traits, phenotypic character that passes from. So what are these? So it has nothing but the characters which are passed from parent to the offspring, from our parents to us. So those are called as traits, basically certain characters. For example, your eyes might be kind of similar to either your mom or dad, or else your facial features kind of be little similar to your either mom or your dad. So that those are nothing but traits. So which are coming from our parental generation to offspring, like I mean to our traits. So he called them as a hereditary rules, which were discussed by him. So in terms of eye variations, these are certain example. In terms of air variation, these are certain examples. So these are coming up with the help of our parents itself. So what hypothesis he made that deck of cards. So if you he just compared with the deck of cards. So if there are three players, the first player is parent, son, daughter, and grandson and daughter. So genes can be shuffled and passed along the generation. So these maybe there are certain genes with so there are certain cards with each player. So imagine them as a genes, and they can be shuffled and accordingly given to the uh, next generation. So blend. So it basically tells us that even this parent has some influence on grandson and granddaughter. Similarly, the grandparents will have some influence here as well because they were shuffled and accordingly certain characters were phenotypically expressed in certain individuals, but the later generation might have certain phenotypic characters of this parent or certain grandparent. That's what this slide means. So he was lucky that he in his monastery there were pea plants. So it had choosing that pea plant was he was lucky enough because it has availability of many variations like different colors of flowers, different seed shape, or different or it can be either in the exile position or terminal position. So all those traits he could study, and there were large number of offspring from each mating. So from each mating he could get large number of offspring so that he could have a defined conclusion out of it. And crossing of them was also easier. That is, pollen and egg bearing organs were easily visible, and uh, they could be easily crossed with each other. So what are the parameters? So initially he chose only true breeding varieties, a purple flowered plant and a purple flowered plant. When it crossed, it always produced a purple flowered plant only because this is dominant. Because they are true breeding, they didn't had a heterozygous form of uh, alleles. And so what the he chose different characters like this. He tried to study flower color, he tried to study flower position, seed color, seed shape, pod color, and stem length as well. So all these characters were studied by Mendel. So initially he studied one characteristics at a time. So he only chose distinct alternative forms of those characters, and he tried to study them by crossing with uh, one feature with the other feature, and tried to understand how is the next generation coming in, and the F1 generation and F2 generations are coming at what ratios. Basically he tried to study that, and by studying that he came up with certain ratios and certain laws of inheritance. So how does the crossing takes place? So he takes the plant, and this is the stamen region, or sorry, this is the carpel region, which is a egg bearing uh, part of the plant, and these are the stamens which produces pollens. So he just used to take these two, cross with each other, and plant it so that accordingly uh, the next generation will come up. So purple was seen to be dominant. So whenever a purple and white flower were crossed with each other, he always got purple flowers and no white flowers, indicating that this is recessive in nature and this is dominant in nature. And this was the F1 generation where all were purple in color, wherein when these were crossed with each other, like the purple one were crossed with all F1 generation were crossed with each other, then he used to found like 705 of purple flowered plant and 224 white colored plants. So he came up with a ratio. This is approximately 3:1 ratio. So he found that when one specific character is crossed, which has one dominant trait and one recessive trait, the ratio comes up with 3:1 as a phenotypic ratio. Now he was trying to study whether in terms of genotypic ratio. So this is the same thing. So in this, he found that he tried to determine the law of segregation. So how segregation takes place. So what is that? So two and form different gametes. So they are so if even if they are together, this P and P, they separate from each other and form different gametes. So that's the reason we use different gametes at a different position here. So because of this law of segregation, even if your using Punnett square method. So this is Punnett square method, and at that time we separate these alleles and then check the inheritance pattern. So he came up with certain ratios like this, where he found that 3.15 is to 1 and 3.14 is to 1 in terms of color, and flower position. And so if you see, approximately it is nothing but 3 is to 1 ratio about dominant versus recessive. In terms of so how do you determine the organism genotype? As I said, the genotype is determined by test cross, wherein a homozygous or heterozygous, if the purple. So we, for example, this specific purple class plant, we don't know that whether it has a this specific genotype or this specific genotype. It is homozygous or heterozygous, we don't know. So what we do is we try and cross it with the uh, homozygous recessive plant wherein a white color is produced. So if a purple cloud flower parent is PP, or if a purple flower parent is P versus capital P and small p. So if you check this, so when it is P and P, when it is capital P and capital P, when these are crossed, so all offsprings are purple in color. But when PP is crossed, like capital P and small p is crossed with the homozygous recessive plant, he used to get half purple and half white in color. So which indicated that if this is the ratio which he observes, then it indicates that this specific plant has a heterozygous combination, capital P and small p. And if this is the combination, like if he observes all are purple in color, then he used to understand that the genotype of this specific plant is capital P and capital P. So test cross actually helps us to determine the organism genotype. And so this was the first lecture when he talked about different laws, law of segregation, law of dominance, and how can we find the genotypic ratio of a genotypic way, how genotype might be of that specific phenotype, whether it be homozygous or heterozygous. Then in the second lecture, he tried to talk about independent assortment. So here, when two traits are studied at a time, he initially thought that maybe two traits are depend. So he hypothesized that the two traits would either be dependent upon each other, and then, for example, uh, in terms of seed color and seed shape. So if yellow color and round shape is dominant, he initially thought that maybe if dominant traits are present, if these two are dependent, like seed shape and seed color, if they are dependent with each other, then if seed color is yellow, then obviously seed shape should be round only, and if seed color is green in color, and then seed shape should be wrinkle only, but that was not the case. So he found that the seed shape used to be different even if the seed color is same, and it actually indicated that both the traits are assorting independently. It is not getting into the next generation depending upon a color or depending upon the seed shape. So both traits individually were assorted and then uh, went into the next generation. So that's what he observed, if you can see here. So a heterozygous, like, so why capital Y is considered as yellow color and small y is considered as green color, whereas capital R is considered as round shape and small r is considered as wrinkled shape. So in this, if everything is recessive, or like small y which is green, and small r which is wrinkled, so it will be wrinkled in color and green in color. Whereas if everything is capital, it should be yellow and round in shape. So when these were crossed with each other, it was found the F1 generation was heterozygous, but heterozygous in nature, but it is yellow and round itself. It is not wrinkled because yellow color and round shape is dominant over green and wrinkled shape. So when they were crossed, these were the different genotypes which can be made up made after crossing these two each other. So when we cross them at each other, so if it was a hypothesis of dependent assortment, so like if there is YR, then it capital YR, it will be capital YR only. If there was a dependent assortment, this might be the ratio. But he actually found that it was independent and he used to get such kind of ratios. So here you can see that even if there is capital Y, there was smaller as well, indicating that even if the color is yellow, the it can be wrinkled shape as well. It is not necessary that if the color is dominant, then the dominant shape will also be there. So the shape is independently inherited, whereas the color is also independently inherited. That was the conclusion from law of independent assortment. And here he tried to study if we study two characters, what is the phenotypic and what is the genotypic ratio. So genotypic ratio here comes up as so he sir has not written here, but the phenotypic ratio, like the way they look, is 9 is to 3 is to 3 is to 1. So along with this Mendel, there were other scientists who were working with different organisms and tried to study the inheritance pattern. Later, these they were the one who got Nobel Prize. And in terms of human traits, so there are some traits which are in terms of us, there are some traits which are followed as per the Mendelian laws, and there are some traits which are not followed as per the Mendelian laws. Those are called as non-Mendelian inheritance pattern, and the one which follow, those are called as Mendelian inheritance pattern. So for that, we generally study pedigree analysis. So what is pedigree analysis? Wherein a family history analysis is done for a particular trait and assembling information in a family tree pattern describing traits of a parent and children across the generation. That is called as a family pedigree. So in this, so basically this analysis helps us like how a particular trait has come up from parent to offsprings. So as an example here, widow speak. So widow speak is a dominant trait, but it is not commonly seen in most of the individual. So, for example, this square indicates male and circle indicates female. And if it sorry, if the person is having widow's peak, it's purple in color, or if a female is having, it is purple in color. So, if there are so first generation which are grandparents, so widow's peak, so W is capital W is dominant, small w is recessive. So for example, a male which who was recessive but had widow's peak, but a female who was sorry in heterozygous form had widow's peak. Similarly, there was other parents who were a female was recessive and male was heterozygous for widow's peak. We can so the individuals who are born in the second generation, the offsprings of this specific parent, you can see that there is a half chances of uh, getting widow's peak and there is half chance of not getting widow's peak. Like that's what the generation was, the next generation. And if, for example, if these two mated which had widow's peak with in an heterozygous form, then there were some individuals with widow's peak and some without widow's peak. So basically, widow's peak is a trait which follows Mendelian genetics. Similarly, and attached earlobe. So here the earlobe is attached, whereas earlobe is not attached. Similarly, like widow's peak, even this trait is a recessive trait generally, but it follows Mendelian genetics. You can accordingly analyze here. So along with the Mendelian laws, there are certain complex inheritance patterns which doesn't follow Mendelian laws. So in terms of dominance, complete dominance was the one which was observed in terms of Mendelian inheritance, wherein occurs when a phenotype of an heterozygote and a dominant homozygote are identical. So a dominant homozygote, for example, was having purple color. The heterozygote was dominance. But there are some characters which also showed incomplete dominance. For example, where a phenotype of two parental varieties, so basically they mix together and there is something different color comes into picture. And codominance is the one whether it affects the phenotypic separately, like all those both characters will be expressed in the uh, next offspring. That is called as codominance. And there are certain, it's not necessary that so this is generally observed similar to that of widow's peak and similar to that of earlobe. The recessive characters are generally are present in more individuals as compared to dominant, but if there is a dominant trait in an individual, so he that person will have chances of getting, so that is because of evolution. Basically whatever is the best suited one, it is actually getting, it is choosing, the natural selection is choosing that. And most of us have such kind of hands and not six fingers here, and we have five fingers. So let's look at certain questions. So if there is a true breeding self-pollinate, all offsprings are of the answer is same type. So true breeding, as we already saw here in this. So if there is two breeding of capital P and capital P, all flowers will be of the same color. So these were of purple colored, and the offspring is also of the same color. So that's what the answer is. So which of the following so touch cross, we already know that it is studied with the recessive plant. So there should be recessive in the option. So this only one option has recessive parent, small a. So the cross should happen with the recessive plant in order to understand the touch cross. So for example, if two alleles leaf color is green and which is capital G, and orange is small g, and if green is dominant, this capital G is dominant, but orange is the most common trait. So it can be common trait can be recessive one, but this is the one which is dominant, capital G. So what is the phenotype of homozygous dominant? So homozygous dominant means this is green is dominant. So homozygous condition, capital G and capital G. If it is capital G and capital G, the color of the leaf should be green itself, right? So that's easy to answer. Okay, so this is one of the questions. So where self-pollination, I hope you all are getting it. Yes, good to go ahead. Yes, Byrunda. Yeah. So if you want something to be explained in much more detail, you can tell me accordingly. Okay. So there is self-pollination. These are heterozygous for flower position and stem length. So flower position के लिए it is a capital A and small a, and stem length it is capital T and small t. So both are heterozygous here because homozygous means capital A होगा या फिर small a it will be. So both are heterozygous. So we have already studied in terms of uh, uh, yellow color and sorry, seed color and seed shape. So instead of seed color and seed shape, here the question is about flower position and stem length. It's the same thing. And what they found out was there were 200 were planted, and how many offerings would you predict to be dwarf with terminal flower, indicating that so we already know that tall is dominant over dwarf. So this is recessive. Even axial position is dominant over terminal flowers. So these both are recessive in nature. And as if you have looked at these ratios in terms of this specific ratio. So if you see here, so capital small y and small r is only once in this whole cross, right? So it indicates that out of 16 possibilities, one possibility is that there can be like the green color which is recessive and wrinkled shape which is recessive. So here these two characters which are asked in the question are recessive, like dwarf and terminal flower. So it is only one out of the 16 combination. So it is 1:16 ratio, but there are 200 plants which are planted, so which were observed. So if you multiply by 200, it comes up as 12.5, which indicates that approximately 12 plants were the one which actually can be the possible that it might have dwarf plant with terminal flowering. So that's how we came up with this answer. And there's one more question which indicates that there is an exile flower position, which is heterozygous in nature, is crossed with a homozygous terminal flower. So here we are studying only one character. So this is nothing but a test cross. If you see here, because we are uh, crossing with the recessive one. So draw Punnett square board. So so these are the two alleles AA and AA. So one allele you will write here because they are independently asserted, and other allele you are write here. Then you just need to write the combination capital A, capital A, small a, small a, and small a, small a. You can clearly see that the ratio is 1:1, like half half. So the genotypic and phenotypic ratio for this is 1:1. So that's what the question was. The other question is, there is in some plants a true breeding red flowered strain gives all pink flowers. So uh, there is a red flowered strain itself, but it gives all pink flowers when crossed with a white flowered strain. So RR indicates red, whereas smaller indicates white, and capital R smaller indicates pink. So you can see here. So even in generally what Mendel saw was capital R smaller was also giving red color. For example, there the option was purple color, right? So capital P was giving purple color, and capital P small p was also giving purple color. But here you see, as per the question, the capital R and small r giving pink color. So this indicates there is incomplete dominance. It is not showing complete dominance over it. So this is a concept of incomplete dominance. So wherein red and white color are getting mixed with each other. So it is not showing, the red is not completely showing its dominance, but just because of the presence of capital R, it mixes with the white and turns out to be pink. So it is not completely dominant, but it is incompletely dominant. So if similarly in terms of seed shape, the round and wrinkled, what will be the ratios of genotype and phenotype in the F1 generation, and resulting the following cross round red, which are true breeding, wrinkled white, what will be the ratios in F2 generation? So if we talk about red and white, let's consider one. Uh, so wait. So let's consider this first as red and white, only the color. So when R and R is giving, it is coming up as pink. So this is nothing but incomplete dominance. That's what I was telling. So red is R and white is small r. And similarly, there was a question for seed shape as well, which is round or wrinkled. So capital S is for round and small s is for wrinkled. So similar to that of the yellow color seed and white and round shaped seed, and green colored seed and wrinkled shaped seed. So similar kind of approach. It's the same question, but here we are studying about the color of the flower, red flower, color of the flower, and shape of the. So here the two traits are color of the flower and shape of the seed, whereas the example which we studied was color of the seed and shape of the seed. It is similar to that. So the ratios will be similar to that of the one we observed over there. So if you can see, the in terms of phenotypic ratios, uh, uh, there it will be little different because it is complete dominance was observed over there, but here we are observing incomplete dominance. So in terms of incomplete dominance, so if you see here, if you solve the question in a similar manner like this, like the one which was for yellow color and yellow color of seed and yellow green color of seed, either of them, or round color seed or wrinkled seed. So if you solve in a similar pattern, so you need to look at this specific uh, genotypes, and accordingly you can come up with the ratios here, because here that is incomplete dominance. If it is in a heterozygous form, if it is in an heterozygous form, it will give you pink color, or else it will give you the different shape as well, because it is incomplete dominance. So because of that, the ratios will change, and this is what the ratios you will come up with if you look at this, all of the genotype of each and every box. Then accordingly, you will come up with this ratio. So there will be round seeds with pink flower, there will be round seed with red flowers, round seeds with white flowers, wrinkled seeds with pink flowers, wrinkled seeds with white flowers, wrinkled seeds with red flower. So that this is what the phenotypic ratio will be, and this is will be the genotypic ratio. So here one more question was: a man had 6 fingers, his wife and his daughters are normal, indicating that wife was having only 5 fingers, whereas daughter was also having 5 fingers, but man had 6 fingers. Remember that the extra digit is dominant trait. So what fraction of this couple children would expected to have extra kids? So you can just randomly use this is the dominant, as it is said that remember that extra digit is dominant. So man was so you can so digits were considered as D here, wherein so as the question itself says, this was dominant in nature and this was female should be recessive. So if you calculate in the like in this way, your child should be DD. So daughter will have an extra digit because this is dominant. But as per the question, daughter didn't had an extra digit. So he should be in heterozygous form and not in homozygous form. Because when you cross with homozygous form, the child will be having this, but daughter was not having uh, any extra digit, even the daughter was normal. So which indicated that male should be in heterozygous form and male should not be in a dominant form. अगर dominant होता तो even daughter should be might have had extra digit, but the daughter doesn't have, indicating that male was heterozygous and female is homozygous. So that's what the answer is. So, inspired by Mendel experiments, so there was an experiment wherein a vinca plant is grown in the farm. So, plant is usually short and bushy, but one growing in the farm is rather long. So, it is not short but long. So, now assume that the height of this plant is controlled by a single gene with short plants, which is capital S, being dominant to long ones. Which of the genotype should be there of your plant? So, what was our plant is usually short and bushy. So since the plant is long, what he found was so being dominant to long ones. So capital S. So he said that capital S is for short plants, and that indicates small s it's for long plants. So as the plant is long, this is a recessive trait. Its genotype should be smallest smallest. Simple to answer. So here they have solved it as well. So our plant was long, which is smallest smallest, and the other plant generally what they used to found was short and bushy. This was 5A question, and in terms of 5B, so could one of the parent of your plant have a short phenotype? If so, what would be the genotype? So because genotype is possible only in the heterozygous form only, because if the parent of that plant had a genotype like this, capital S smallest and smallest, they should have still produced a progeny with smallest genotype. So that's the reason it should be cap in an heterozygous form. So you can look at the answer here. So it should be in the heterozygous form itself. Then only heterozygous will be half will be short and half will be long. then only it will be able to produce. If that was not the case, then all would have been short plant only. There wouldn't have been any long plant. So there might be one small s in the plant. So that's what they were asking, right? So what would be the genotype of that parent? If could one of the parent of your plant had short phenotype? So there should be one small s only if that was the case, and then only a long plant will be produced. If there is no long s in the other parent, there like small s in the other parent, long plants won't be produced. So that's what the uh, solution talks about. So these were the deviations as we were talking about. So red flower and white flower comes up with pink flower. This is called as incomplete dominance. So now the genotypic ratio here, the phenotypic ratio itself is 1 is to 2 is to 1. There are two pink, one red, and one uh, white. There is something other concept which is albinism, homozygous in nature. So if a parent is a carrier, heterozygous, but themselves have normal phenotype, what would be the possibility of the carrier children? So what do you think? If two heterozygous are matched, so if two heterozygous are matched with crossed with each other, there is only one possibility of having recessive plant, a recessive individual. So these are normal because it is only expressed, albinism is only expressed in a homozygous recessive genotype. So if they both were heterozygous in nature, but they were normal, so that is only if you cross them. So if you write the ja- phenotype of that like genotype because they are independently assorted, so genotype of that and genotype of this male and female, then if you make a Punnett square, then you can clearly see that there can be only one possibility happening. So 1 is to 2 is to 1, or else all are normal, so here the ratio is 3 is to 1. Genotypic is 1 is to 2 is to 1. So here, in terms of incomplete dominance, what happens is the phenotypic as well as genotypic ratio is same, 1 is to 2 is to 1. Whereas here, again, we are talking about the Mendelian pattern, wherein the genotypic is 1 is to 2 is to 1, but phenotypic is 3 is to 1. In terms of blood grouping as well, we have four forms of blood groups A, B, and O. So it depends upon the carbohydrates present on the uh, RBCs, and it can be either in the homozygous form. So A blood group can be in the homozygous form or in the heterozygous form. If the recessive part comes into picture, then it is O blood group, small I. And if A and B both come together, both carbohydrates are present, and it is giving A and B. So there was a demonstration of it as well. So an A and B blood groups are dominant over O blood groups, and A and B genes are codominant. So they can exist together as well. So this is called as codominant property. And generally, if a recessive part is present, like O part, A is dominant over the O blood group. So that's what it is written. A and B blood groups are dominant over O blood group. So if a person is having O blood group, then both recessive should be together. Then only a person can have a O blood group. Then you might have heard about positive and negative also. So there is a way of testing a person's blood group. So if you are using anti-A, that anti-A will bind with these carbohydrates, and accordingly agglutination will be seen. So if a person is uh, A blood group, then anti-A, the agglutination will be seen. That's the reason a person is having A blood group. But indicating positive or negative, so there is an anti-D which is RH factor. So if that RH factor is also expressed, then agglutination will take place, indicating that this individual is A positive and this individual is A negative because anti-A shows agglutination. Similarly, for O, so there are no antibodies like antigens on O, right? So there will be no agglutination either with A, either with B. So there is no agglutination A with either A or either B. Whereas anti-D just talks about positive and negative. If there is agglutination, then it is positive. If there is no agglutination, it is negative. So that's how we can predict the blood group. If a person is having B blood means he possesses which antigen and antibody. So B है मतलब B antigen होगा on the red blood cells. It is common sense. So, what kind of inheritance pattern or alleles exhibited by ABO blood group? So there is codominance as well because A and B can coexist together. So there is codominance and there are multiple alleles. There is IA, there is IB, there is IO, there is IAB, and so there are multiple. So it is a concept of multiple alleles as well as codominance. So that's the reason the answer is both A and B. So this question is also good. So there is there are three individual, three donors, and we want a uh, blood group A with a positive. A positive, we want an individual because that is the individual who has infected, and there are three donors, and this is how the three donors are. And we want to know. So if we want to transform the blood from these individuals to him. So we need to choose. So which is observed in three cases, indicating that all are having A antigen. So all can be part of it, but we need to see whether they agglutinate with B also and RH positive as well. That's only then we can give the blood to. We can choose that individual for this blood transfusion. So there was X individual who had agglutination with B as well, but indicating that he is AB but not with anti-D, indicating he is AB negative. So this donor X is AB negative, whereas donor Y shows agglutination with anti-D alone. So indicating that this Y individual is A positive. So A positive, and we want A positive itself. So we will obviously choosing donor Y. Y indicating and either with B indicating he is A negative. So Z was the one who is A negative, Y was A positive, X was AB negative. So we wanted A positive, so we will choose Y as an individual. So which statement is true in terms of ABO? So both allele, A allele and B allele are dominant over O allele. Yes, that's true. So in terms of a person having AB blood group, should be able to receive blood from donors of which of the following? So AB blood group has, so if you see here, AB blood groups has both the uh, uh, carbohydrates and if you see in terms of AB blood group, it agglutinates with everything, correct? If it is positive, it will agglutinate with everything. If it is negative, it will agglutinate with A and B only. So here the question is, a person having AB blood group should be able to receive the blood from donors, which of the following blood group. So AB blood group, if you all know, he is the universal acceptor and O is the universal donor. So you can accept blood group from anyone. So either it be B, AB, OB or AO. Anybody can donate the blood to him. He can receive the blood from them. So it is all of the above. So here, a man with A blood group marries a woman with type B, so IA and IB. Their child has O blood group. What are the genotypes of these individuals? So if it is O, indicating that A and B were not dominant, they were recessive because their child had O blood group. So indicating that these were recessive only, then only these two I can go in a 20. There will be a 25% chance that uh, O blood group can also be there in the offspring. If they were not, if they were dominant, like I capital A and I capital A and I capital B and I capital B, so there would be no chance that an individual, the offspring, will have O blood group. So it's obvious. So what are the other genotypes and what is the frequency? So when you cross it, so IA, IB, then IA and I, then IB and I here, then small I and small I. So you can clearly see each ratio is about 25%. So 1:4 is the answer for expected of frequency, and the genotype will be I and I. So imagine a newly discovered recessive inherited disease is expressed only in individuals with type O blood. So the person who is having O blood group had a certain disease. Although the disease and blood group are independently inherited, a normal man with type A so, and a normal woman with type B already had one child with the disease, so they might be recessive because then only O blood group will go. The woman is non-pregnant, is now pregnant for a second time. So what is the probability that the second child will also have the disease? So assume that both parents are heterozygous. So we already gave the uh, genotype of this and with that causes the disease. So here instead of using IA, IB which creates certain confusion, the answer is solved by using capital A, capital B for blood groups of parents and small a and small b which is associated with disease and D indicates a disease gene. So what they have done is heterozygous A blood group, heterozygous B blood group, and the disease pattern. And for diseases, the dominant if it is capital D and it is dominant and small d is recessive. But what they have said that it is recessively inherited. So indicates that if there is small d and small d, then only a person will be having that disease with the O blood group. So there is O blood group as well as recessive inheritance pattern. So recessive indicating that if a person disease is small d and small d, then only the diseases will be there, but only in O blood group. So indicating that small a, small b should be there, then only a person will be having O blood group. Because even if capital A, capital B is there, so here he will be not be having disease because of capital D. Here no disease because of capital B because it is B blood group. Even he is B blood group individual. So only O blood group and with recessive pattern. So indicating that there is only one chance out of 16. So the answer should be 1 by 16. So this was the summary of the that lecture. And then what's the time? Okay. So do you all want a break of 2 minutes? Maybe. Yes. Are you getting it till now? You can answer in chat box. Yes. Yeah. Okay. Do you want anything to be explained in detail, anything, or then you can let me know accordingly. So let's take a break of maybe 2 minutes, then we can start again. Okay, we'll start again. Okay, so I hope I'm on my mic. Okay, so in terms of probability, um, so in the next lecture, sir has tried to use certain probability like logics to understand. So it is easier, it will be easier to calculate how how a specific genotype will come into picture, or how a specific phenotype will come into picture using probability. So is always between 0 to 1. So you can accordingly use either it will be half or so if two heterozygous are crossed with each other, then half will be having dominant trait, half will be having recessive trait. Then accordingly, if they are crossed with each other, everyone will be having a 1 by 4 chance of getting specific trait of that. So similarly for independent assortment as well, wherein seed color and seed shape was used. So what how we can use probability in terms here is, so instead of making this whole Punnett board, we can use probability by considering each trait separately. So if you consider this yellow colored or green colored trait here and red colored trait here, uh, in terms of finding out the answers. So we what instead of, so this is a little complicated to make, right? Like making the whole Punnett square board using two different traits. So instead of so what we can do, we can use one trait separately and other trait separately. So when you cross these traits, heterozygous RR. So you can take one trait and cross with the same trait. You can take the other trait and cross with the same. Then you will come up with these certain ratios, 1 is to 4, 1 is to 2, and 1 is to 4, because these are having 1 is to 4 chances, 1 out of 4. This is 1 out of 4, whereas these two are basically 2 out of 4, which is nothing but 1 by 2. So these were the ratios. So when you uh, take this with one specific trait, like one specific trait, for example, round shape, which was 1 is to 4. Now, how many round shape will be there with either yellow color or in the heterozygous form or in the recessive form, which is green in color? So so we have individual ratios. We can just multiply them here and we can come up with the answer. So 1 is to 4 uh, multiplied by 1 is to 4, which is 1 is to 16, 1 is to 4 multiplied by 1 is to 2, which is 1 is to 8. So we can come up with these ratios for this specific phenotype just by using probability instead of making such Punnett square board. So that was is explained here as well. So if you want to find a probability of capital Y, capital R in the F2 generation, you can simply use this specific ratio, capital Y, which was 1/4 in this specific and capital R in this specific Punnett square board. So 1 is to 4 multiplied by 1 is to 4, which is nothing but 1 is to 16. So you can directly get the answer by using probability instead of making such whole board and then understanding how many will be of that specific phenotype, or how many will be of that specific genotype. So instead of doing that, you can use this specific probability method and come up with the answer in a easier way and in a sooner way as well. So that's what is talked about over there. So what is the probability of having an albino child if both parents are heterozygous? So we have already solved that. So there will be 1 out of 4 chances. It will be 1 by 4. Similarly, for a dwarfism, which is one of the disease which is dominant allele. So what is the probability of having a child with disease if one parent is homozygous. So one parent is homozygous recessive. So this is a dominant allele and homozygous recessive. So one parent is small d and other parent is heterozygous capital D small d. So if you just make a Punnett square board out of that, so you can see that there is half chances of being dwarf. So what is the probability that the child will be with the disease? So there is half possibility of dwarf and half will be normal. So half will be dwarf. So that's what the solution tells about. So here we are considering a dihybrid cross with heterozygous form, and what will be the probability that the genotype of the offspring will be this kind. So as using probability itself, so when you cross these specific capital Y small y with capital Y small y, you will get these ratios 1:4, 1:2, and 1:4, right? So now what you need is capital Y and small y. So capital Y and small y, capital Y and small y is half possibility, right? Now there is small r as well. So you can similarly solve this as well. Small capital R, small r, capital R, small r. So there will be 1/4 possibility of having small r and small r. So if you write the same kind of ratios with capital R and small r, you will end up with small r 1/4 way. So you are just multiplying this half into 1 by 2 into 1 by 4, you will end up as 1 by 8. Simple. If you're not getting it, you can let me know. Similarly, for tri-hybrid cross, assuming independent assortment of all three genes. So you can individually, for each allele, you need to make such ratios. So capital A, and capital A are crossed. If they are crossed, then these are the ratios you will end up. If capital B, small b, capital B are crossed, then same ratios will be coming up, and capital C, small c will also be giving you the same ratios like capital B, small capital B, small a, capital B, small a, small a, small b, small b. That's what it will be for B as well as for C and as well as for A. Right? If you individually solve them, so what are the probability that F2 offspring will have the following genotype? So these are the following genotype. You need to form the probability for each kind of genotypes. So for all small b, or small c, so everything will be 1:4 chances. So 1:4 into 1:4 into 1:4 will give you this answer. But for this, there will be 1 by 2 multiplied by 1 by 2 multiplied by 1 by 2. So if you do 1 by 4, 1 by 4, and 1 by 4. If you multiply, you will get this answer. So that's what you will get as an answer. So similarly, you can solve for this, and that's how you can end up getting these answers. I hope it is easy to understand. If not, you can let me know. And similarly, there was one more question wherein each character is controlled by independent assorting. One is dominant and other is recessive. If a plant is heterozygous, so this is heterozygous for all three characters. So that's what X, Y, and Z is considered. All three characters, it is heterozygous in nature. So what is the proportion of offspring would you expect in terms of probability to be homozygous for the recessive traits? So homozygous means for the recessive in the sense small x, small y, small z. So if you have seen here for small like recessive trait, it is 1:4. So you just need to multiply 1:4, 1:4, 1:4, you'll get the answer. So basically, probability will help you to get such answers in a quicker manner instead of making the whole Punnett square board and then coming up with the answer. So there was one more question wherein inherited diseases. If a woman and her husband were both carriers, so both carriers in the sense both are heterozygous and they have children, what is the probability of each of the following? So similar, there are four questions by A, B, C, D. So we need to come up with, so all three children are normal phenotype. So normal is 3:4. So the answer will be for three children, 3:4 into 3:4 into 3:4. So this is the chances, basically the probability. If one or more of the children are having diseases. So there is so only one is having diseases out of these three children. So if you just minus that, so you will get the answer for the second one. All three children have the disease. So there is only 1/4 chance of having a disease. So 1 is to 4 into 1 is to 4 into 1 is to 4 for all three children. So there is 1 is to 64 is the probability of that. So at least one child is phenotypically normal. Note that the probability of all outcomes always add up to 1. So if all is adding up to 1, so we need, so 1 minus 1 by 63 will come up as 63 by 63. So it is all, at least one child is phenotypically normal. So this is the probability of that. The phenotypical normal because there is only rare chances that it would be recessive and it would be having disease. So basically there is a hint here that it is always adds up to 1. So that will help you to uh, understand that it should be minused by subtracted by 1. So three characters. These are the three characters of plant P, 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 Y, and I. What fraction of offsprings are predicted to be homozygous recessive for at least two of these three characters? So uh, initial questions were all the characters should be recessive trait. But now so here all the characters X, X, Y, Y, Z all were recessive trait. But now the question is at least two of them with the recessive trait. So here you can see these two are with recessive trait. Here at least two means two or more than two. So all three are in recessive trait. Here P and I are in recessive trait. Here Y and I are in recessive trait, and here P and I are in recessive trait. So these are the following possibilities. So now we want to fractions we want to determine of the same. So instead of, so similarly you will so capital P small p and small m small p, you will cross them. When you cross them you will end up with this equation, these possibilities. Similarly capital Y capital Y small I, so you will end up with these ratios, and similarly capital I small I small I small I, you will end up with this ratio when they are crossed. So now, according to these possibilities which you already made, so you will just multiply accordingly. So small p is half chances, small y is again 1/4 chances, and capital I small I is half chances. So half into 1 by 4 into 1 by 2. So that's how you will be able to solve them in a much more quicker and easier manner. So this was the summary of that lecture and conclusion. So you can, for sums, basically you can look this modified from Campbell book itself, and there is the reference is from Campbell biology book. So you can use this specific book for understanding more questions. That was about the fourth lecture. Then in the last lecture, there was a talk by Dr. Jayanti Shastri where she talked about challenges and opportunities related to infectious diseases. So how samples are collected, how antibiotic resistance builds up in the body. So he explained about all of this. I'll be just giving overview of it. So what are the different challenges if there is a bacterial infection, if there is a viral infection, if there is an antibiotic resistance being built up? So what kind of uh, detection techniques are used to understand what kind of disease a person is having? There are serological methods wherein antigen antibody is used. There are different methods to isolate bacteria, virus, etc., and their genome detection as well as antigen detection methods. These are called as direct methods. And when you are using antigen antibody for detecting this antigen or genome of it, it is called as indirect methods. Then there were some clinical manifest which were shown by her about dengue and as well as about malaria. So in terms of challenges, so we don't know like which individual will progress to severe disease in dengue and malaria. So there are certain genetic predisposition or host responses being seen. So these are certain opportunities coming up. So how we can analyze? So there are different technologies for diagnosing. That is nanotechnology is used in diagnosis. There are uh, people are studying with coming up with certain biomarkers which will help us to understand disease progression. So biomarker in the sense, for example, in the blood itself, there will be certain biomolecules, maybe a carbohydrate or amino acid or certain lipid concentration, which changes in a disease individual as compared to that of a normal individual. So people are trying to find in a disease individual what kind of biomarkers are expressed more, are down regulated or up regulated as compared to that of normal. So people are coming with certain disease progression biomarkers as well. And currently there are many like SARS-CoV, Ebola, all those are emerging infections, and how people are trying to come up, like trying to solve these issues as well. So she coming up with certain technology for diagnosis as well as coming up with certain treatment approaches. So how that is helping us, that also was explained by her. And lastly, she gave certain words of caution. So most known protein structures have been worked out, but tied by network of chemical bonds. But in reality, proteins are very much flexible, but that is much harder to deal with. So we cannot study them also, but in nature, that's how proteins are. It is not stable enough, and in order to study the structure, we make it tied by networks by chemical bonds in order to study the structure of it. But in nature, they are not. So even by studying that in nature, they might behave in a different way. So that was the one in terms of lectures. So now we'll come up with certain previous year assignment questions, and we'll check whether if we can solve them. I hope it was a good revision for you all. For you, like you got a whole five lectures in a crisp manner, and you already know how, as sir has already gave n number of examples for solving questions related to genetics. I think this previous year assignment questions will be easier to deal with for you all. Okay, so what is the possible genotype in context to the blood group? So a man has a O positive blood group, means this is a specific genotype of it. And if a woman have a girl child, so they are having a girl child. So it is nothing to do with a boy child or a girl child, but it is with the blood group. So if a woman is having B positive blood group, so it can be either IB or it can be either IBI in a recessive form or in a homozygous form. But he is O, that man is having O blood group, indicating that it should be in this specific form itself. So what could be the possible genotype in context of blood group? So what do you think? What would be the possible genotype of a girl child which is born? What do you think? So one is recessive, and this we don't know whether it is in a homozygous form or an heterozygous form. So, so if you look at the options, there is capital B and capital B, capital B and capital B. So this is not possible at all, because the genotype of this individual is small I and small I. So there is no B here. So other allele, so one allele will be coming from one parent, other allele will be coming from another parent, right? So this, if even if one B is coming from this parent, the other B cannot be from this parent, because the individual's genotype is this. So option first and option second is for sure not possible. So we need to check whether uh, option B is possible or only option C is possible. So here, even if the individual is homozygous or heterozygous, then IB will be a possibility uh, here. So because even if you take this as an option, IB, so IBIO will obviously be there, and so that's what only be formed, IB with IO. So if only IBIO was the form, and this So, but they are saying that there is a possibility of small I as well, which is also written as IO and IO. So that is also a possibility if this individual is heterozygous, because this specific allele will cross with this allele or this allele. So it will form small I. So option B should be the answer. Yes, that's what the answer is. I hope you are getting it. Where is the, I think I got logged out. Wait a minute. I'll share the screen. Okay, so this is the next question wherein Suraj observed the following three different plants in his garden: plant A, plant B, and plant C. Wherein, in terms of plant B, when a white plant was crossed with black plant, it gave gray colored plant. In terms of plant B, when a white flowered plant was crossed with black plant, it gave white plant itself. Whereas, in terms of plant C, when a black plant was crossed with plant C, it gave both the colors, black as well as white. So this is indicating codominance because both are being observed. This is indicating incomplete dominance, indicating that both are getting mixed and giving a specific color. Whereas this is complete dominance. So this is complete, this is incomplete, this is codominance. So let's look at the options. So we need to find a correct statement. So plant C follows Mendelian inheritance. So in Mendelian inheritance is only this. These are non-Mendelian one. So plant B does not follow the Mendelian inheritance pattern. Plant B, no, that's not the case. Plant A follows the Mendelian inheritance pattern. Plant A also doesn't follow. It is incomplete dominance. And plant B follows Mendelian inheritance. Yes, plant B follows Mendelian inheritance. Two plant, one is tall, one is short, and so how much percent of gray short trees would you expect in F1 and F2 generation respectively? So if you see if there is a dominant allele G present, then it is green in color. But what you want to, and if so, if it will be either capital G, then it will have a green color, or if even if so one capital, and even if it is in a heterozygous form, it will be gray in color. What we want to find is how much percentage of gray short trees. So short will be only present if it is in a recessive form, small t, right? And gray color will be formed if when it is in heterozygous form. So how much is the percent? So we need to find in terms of percentage. So if you solve this, you will end up with 1:4 and 1:4. Like out of four possibilities, there will be, if you individually solve them, it will be 25%. So in F1 generation as well as in F2 generation. Okay, so we need to read these four statements and we'll need to come up with which one is correct. So statement one says that if two genes segregate to resultant gametes, one one in each gamete, two alleles of a gene segregate to resultant gametes, both in each gamete. So it is resultant gametes, one in each gamete, two alleles of a gene segregate to result and gametes, one in each gamete. So here two alleles are the one, whether here people talked about two genes. Then the statement 4 is where four genes segregate to result in gametes and one in each gamete. So which one feel that? So in terms of Mendelian, we talk about alleles and not genes. So it should be either statement 2 or either statement 3. What is understand by this is two alleles of a gene segregate to result in gametes. So both in each gamete or one in each gamete. So you need to come up with an answer. So it is statement 3. So obviously they are independently assorted. So both are in the not in the each in the same gamete. So one in each gamete, whether there are two alleles, each gamete will be differently assorted. So then consider a dihybrid cross between a black-eyed uh, so there is a black-eyed and brown-furred guinea pig with a red-eyed and white-furred guinea pig producing a black-eyed and a brown-colored fur guinea pig. So what would be the genetic constituent with which offspring should be mated to form a back cross? So what is a back cross? So back cross is something which happens with the recessive parent. So this should be crossed with this. So what is the genetic constituent with which of the offspring should be mated to perform a back cross? So that back cross should be the one with the dominant one. So that's the reason the answer is D here. So the question is with which offspring should be mated to perform a back cross? So back cross is done with the dominant one, which is black-eyed brown-furred one. So which is this specific genotype. Consider a dihybrid cross between black-eyed bodied and red-eyed fly. So this is black-eyed bodied and red-eyed. This is black-bodied and red-eyed fly and a black-bodied. So the body is black in color, but a green-eyed fly producing four offspring. Okay. To determine the genotypic constituent of the other parent. So now we want to determine the genotype of this parent. What would it be? So if this is the offspring pattern. So black is dominant, gray is recessive. So if it was forming recessive, so this is uh, so if this they can form recessive pattern, but so this might be having small b as well. Then only small b and small b will go into the next generation. So this is recessive for sure in terms of black colored or gray colored. So the options should be either first one or third one or fourth one. So there is no chance that it would be capital B, capital B. If it would have been capital B, then this was not a possibility, right? Small b will not be in the next generation because one B comes from this parent and one B will come from this parent. So it should have small b as well. So it can be either first option, third option, or fourth option. So let's look at the RR. Uh, so capital R, small r. So if you can see, there is a possibility of small r as well. So indicating that if this is an R, this also has small r with it. And there was small r here as well with this specific. So capital B, and smaller. So it should have small r in its genotype as well. So if you look at the genotype of it, it should be capital B, small b, and smaller because that's what the possibility will be. So capital B and capital B and small b will be the genotype of this with smaller and smaller, because smaller and smaller in two bodies is possible only if this specific individual is having smaller and smaller. So looking at so the answer is already given here. So accordingly, using this specific genotypes, you can just add it here and cross it and determine whether those specific offsprings can be produced or not. That's how you can come up with this answer. So in terms of this question, so there was a red colored allele which is not completely dominant over white colored allele. So red color is there, but it's not completely dominant, encoding for fur in the guinea pig. So there is red colored allele is not completely dominant over white colored allele, encoding for fur color in guinea pig. If a red colored guinea pig is crossed with a white colored guinea pig, so the red colored guinea pig is crossed with a white colored guinea pig, it produces pink colored guinea pig. So now, so if two such pink colored, so all heterozygous are crossed, what is the expected offspring? So if a pink and a pink is crossed, so it is similar to that of a red colored flower and a white colored flower. When they were crossed, it was forming a pink colored flower. So if we cross both pink color flower, so what would be the like phenotypic ratio which will be observed? So if you try and solve that using that specific, let's go to that specific pink and so you can directly get the answer basically. Okay, so if you see, there will be one dominant color which is red, there will be one recessive color which is white, and there will be two incomplete dominance color pink. So this is how the ratio was. So let's see in terms of that fur color question. So it is similar to that of flower color which we dealt with in the lecture. If you look at this question, yeah, so there were two pink, so there will be two pink, there was one red and there was one white, right? So one red, one white, and two pink. The answer should be fourth one, D, answer D. Yeah. So you can solve that using that specific flower question. Eighth question is, consider a man which has six digits. If he is married to a woman having, so a normal one, and man was six digit and this was woman was five digit. After genetic counseling, the couple was informed that the child will have six digit. What would you feel the genetic constituent of the man respective of the digit? So even this question can be solved using that. So it indicated that the child will have six digit. So let's look at that question. Okay. So now the question says that the child will have six digit. Here the question which we solved earlier was, so it had a normal daughter. It didn't have the six digit. So this was the male's uh, genotype. But here the question says that the one which we are solving right now, that question says that the child will have six digit, right? So this clearly indicates that the male genotype would be completely dominant. So that like capital D and capital D. So that's what the answer will be in terms of this question. So what would be the genetic constituent of a man? It should be capital D and capital D, because that specific child has a six digit. That's what the counselor was saying, because this was the genotype of the man. Okay, so consider the following green colored maize plant is crossed with a brown colored maize plant. So there is a green colored maize plant crossed with a brown colored maize plant producing. So there, so these are the offsprings which are produced. So green color is dominant over brown color. Uh, Follows Mendelian inheritance pattern. So which of the following statement is correct? So green is dominant, brown is recessive. So so a green colored maize was crossed with brown. So a complete dominant capital A and small a were crossed, and these were the results. So which of the following statement is correct in this concept? Both parents are homozygous. So basically, it says that it follows Mendelian inheritance. So it should be homozygous for one parent at least. That is, brown color should be homozygous. But in terms of green color, we don't know whether that parent was homozygous or heterozygous. But if you look at the offsprings, there were two chances of offspring having uh, uh, brown color, indicating that one of the parents should be heterozygous and both parents cannot be uh, in a heterozygous condition. So one is heterozygous, one is homozygous if you look at the answer itself. So the answer is one parent is heterozygous, whether one other one is homozygous. The other one homozygous is for sure for brown color because green is dominant over brown and it follows Mendelian inheritance. So it will only express brown color when it is in a recessive state, small a. And the green color can be expressed either when it is homozygous state, capital A, or capital A small a. That can be either of the way. But if you look at the offsprings, there were two half of the chances offsprings were of brown color, indicating that this green color offspring should be in a heterozygous form. So if these were the trait, so now consider a brown colored maize plant is crossed with another maize plant producing these specific offspring. So brown colored is mated in the sense that brown will be having uh, uh, homozygous recessive trait that is small a. So if the maize plants are crossed among themselves, these maize plants, if these like the one which are green, so this should be heterozygous in nature. What is the expected genotypic ratio of the organism? So heterozygous crossed with heterozygous, what is the genotypic ratio? It is 1 is to 2 is to 1. So if that's what the answer is. So if you are having any kind of doubts and if you want to repeat any of the question, we can discuss that. Yes, you have raised your hand. Tell me. You can write it in chat box if you want. Are you having any doubts? Do you want me to repeat anything? Any question? Yes, all good. Okay, so these were the few questions which we discussed, and these were certain questions which we discussed, even with the certain concepts which we tried to clear. And so it was all about Mendel laws and deviations from Mendel laws, and what kind of questions can be asked with this using this, like how Mendel came up with these equations or basically these ratios by looking at P plant, then using them itself, how basically using those laws or with Mendelian genetics or non-Mendelian genetics, how we can understand whether what kind of gen genotype or what kind of inheritance we will be having from our parents. So now we can understand all of those stuffs because of this specific laws which were created by Mendel. Yes. Where are you from? Like you are doing what? Which course? Okay. I think everything is done. Maybe we can wait for like 5-10 minutes more if someone joins and ask any questions, or else we can end the lecture.